So the lipid metabolism is an essential part of the cells to make and break lipids. Okay. And we need to understand what are lipids and how they help. It's a very diverse group of molecules. You have fats, you have phospholipids, and you have steroids and carotenoids. So all of you may remember how these molecules are, what are they are made up of. So this glycerol is the basic building block, plus 1, 2, or 3 fatty acids. That's what will make a mono, di, or a triglyceride. So you have this 3-carbon molecule. Glycerol is connected to an ester bond, and then you have this fatty acid. They are connected like this. So that is how you have the fats made. And phospholipids has glycerol plus 2 fatty acids plus a phosphate. Okay. And they may or may not have a chemical group. In addition to the phosphate, they have another chemical group. So that is what makes a phospholipid. If you remember, the glycerol is what is connecting everything, and it contains a phosphate, and these one contain the fatty acid side chain. Okay, so this has this ester bond, and this is what looks like this phospholipid. So that is the phosphate, and these are these fatty acid tails. Okay. Steroids, on the other hand, they have 4 fused carbon ring structures. They are very typical of the steroids, with or without any additional chemical groups. This is a generic structure of steroid, 4 fused carbon rings. So if you look at these structures, they are mainly hydrocarbons, and the same with the carotenoids. They have 2 ring structures, and they are connected by hydrocarbon, and that's what a carotenoid looks like. So that's a kind of an overview of what are the different types of lipids. Now I'm going to get into how these are made. So the second part, let's get into overview. The second part is lipid synthesis. So your starting material is from carbohydrates. They undergo glycolysis. And acetyl-CoA formation. The acetyl-CoA is a 2-carbon molecule, and so this acetyl-CoA is the one that's like a place an intersection between lipids and this. Okay, so any excess acetyl-CoA more than what is needed for Krebs cycle. for Krebs cycle. So that is what is converted to fatty acids. That's the main overview that you need to know by the enzyme complex called fatty acid synthase. That's a level of detail that you need to know. That's for the fatty acid. And the glycerol comes from glyceraldehyde 3-phosphate out of glycolysis. So that's what is modified, and these two combined together make the well. So the phospholipid is nothing but diglyceride that's coming from the above attached to a phosphate. That's where that comes in. It's a phospholipid, okay? Plus minus a chemical group. Okay. So the next one, how do you make steroids? The steroid is it's an essential molecule for the cell membrane, without which there's a fluidity is not maintained. So there is an enzyme called HMG-CoA reductase. Okay, is the key regulatory enzyme. So many of the statins that we consume that are used to inhibit this enzyme to make cholesterol in the liver. Okay, so the HMG-CoA reductase. And carotenoids, on the other hand, so they are made up of isoprene units. Isoprene is a 5-carbon molecule, and that's a level of detail you need to know. Okay. So the lipid breakdown is the next one we want to understand. So lipid breakdown is the first part is fats are broken down by a process called beta oxidation, where they will be converted to acetyl-CoA. Then that acetyl-CoA can go to Krebs cycle and make more ATP. So fats store more energy per gram than carbohydrate and protein combined together. Each of them make only 4 grams per food calories per gram, whereas fats can make up to 9 food calories per gram. Okay, so the cholesterol that travel in the blood as LDL cholesterol or HDL cholesterol, so they are taken in by a process we have seen before called receptor-mediated endocytosis. And broken down. There are enzymes that will break down the carbon rings. That's how, if you don't have enough receptors to take this cholesterol, you tend to have high blood cholesterol level. Okay, so that's based on some genetic aspects. And the others, carotenoids, so they would be broken down into isoprene units to either carbon skeleton or isoprene units, which is a 5-carbon molecule. And there are enzymes called lipase is a common enzyme that breaks down the lipids. Okay, that's it. That's like a big picture, high level about lipid metabolism. And there are a lot of practical applications of this about how the body regulates the lipids. Like even if you eat celery all the time or rice all the time, body will make cholesterol, even though celery and rice don't have cholesterol. The body needs cholesterol. So that's why it's all important. And everything comes from carbohydrate, carbon, hydrogen. That's the main skeleton that is used to build the lipids. Okay, we are done with this lecture.